In this video, we're going to look at the chemiosmotic theory we're going to look at electron transport and ATP synthesis, inhibition, and thermogenesis. So the chemiosmotic theory we discussed this a little bit before. So it's the dependence of electron transport on ATP synthesis. Right, so that the flow of electrons through electron transport is going to generate a proton gradient which is both a chemical gradient when we look at the change in pH and an electrical gradient when you look at the change in charge. And this gradient is going to drive ATP synthesis. If you watch the other AK lectures on the ATP complex, what you'll see is that ATP can be synthesized without the gradient, but in order for its release into the matrix, we need protons to flow. Okay, so release requires this gradient. So, if we do an experiment, we're going to look at inhibition of electron transport and ATP synthesis right now. So if we do an experiment where we have isolated mitochondria and we add an oxidizable substrate, such as succinate, and then we also add ADP and inorganic phosphate. What we can do is we can look at O2 consumption and ATP synthesis, and we can change the parameters to see how we can affect this process. So if we look at the first graph in A, what you can see, the blue line is showing oxygen consumption. So that we have mitochondria, right? We add a little bit of ADP and inorganic phosphate, and some oxygen is consumed. But you can see it's happening at a very slow rate because we don't have electron transport, because we have no source of NADH and FADH2. Then, if at some time we add succinate, which is an oxidizable substrate that we can get reducing equivalents from, such as NADH and FADH2, you can see you get an increase in O2 consumption because now we have electron transport. We have reduced electron carriers. We have NADH and FADH2. If you look at the red line is monitoring ATP synthesis. And again, as we've demonstrated, electron transport and ATP synthesis are coupled by that chemiosmotic theory. So if you look at the beginning, right, when you just add ADP and inorganic phosphate, you have a little bit slow increase in ATP synthesis. But once you add succinate, there's a slight lag, and there's a lag where you're making the NADH and the FADH2. Now that these electron carriers have transferred their electrons, we see an increase in ATP synthesis. In figure B, right, what we're going to do is we're going to add succinate first. So succinate is our oxidizable substrate. So you can see we have a slight consumption of oxygen, but not very much, okay, because we need to regenerate NAD plus and FAD. All right, so that only happens when our protons flow back through the gradient. All right. So even though we may transfer electrons to oxygen, we're not going to consume a lot of oxygen because the process is not coupled. Now once we add ADP and inorganic phosphate, protons can flow down their gradient, driving electron transport forward, and you get an increase in O2 consumption, and we get an increase in ATP consumption. And again, you can see that lag here. All right, that's followed by ATP synthesis, because first we have to have electron transport, followed by ATP synthesis. Here in this slide, we're looking at the inhibitors of cyanide and 2,4-dinitrophenol. Now, these are two different types of inhibitors. All right, so in our experiment, we have succinate and we have ADP and inorganic phosphate. So we have both the increase in O2 consumption and an increase in ATP synthesis. Cyanide is going to inhibit complex 4 of electron transport. So that is going to decrease oxygen consumption, and it will shut off ATP synthesis because we are no longer generating our proton gradient. So it turns off electron transport because it inhibits complex 4. And then in turn it will shut off ATP synthesis, because if you remember, complex 4 pumps protons out. It pumps 4 protons out. Dinitrophenol is an uncoupler. Okay. What this will do is this will uncouple electron transport and ATP synthesis. Right. Because we still have cyanide present, which is inhibiting complex 4, we do not get an increase in oxygen consumption. And we still have no ATP synthesis. Remember, in order to synthesize ATP, or essentially release the ATP, we need that proton gradient. This slide is showing you how 2,4-dinitrophenol is used as an uncoupler. So 2,4-dinitrophenol can pass through the inner mitochondrial membrane. When it does this, okay, it brings a hydrogen with it. This hydrogen can dissociate into the matrix side, thereby dissipating the proton gradient. Remember, we generate our proton gradient by pumping hydrogens out. So if we bring one in without the use of ATP synthase, right, we're going to dissipate that gradient. And instead of using the energy of this diffusion to make ATP, the energy is lost as heat. We're going to see this mechanism in hibernating animals when we look at thermogenesis. In this slide, you can see several other inhibitors of electron transport. So here, you need to know rotenone. Rotenone is used as rat poison. Rotenone is going to inhibit complex 1, so it also inhibits proton pumping. Antimyosin, you need to know, is going to inhibit complex 3. And then you need to know azide, cyanide, and carbon monoxide all inhibit complex 4. So all of these will stop proton pumping, which disrupts the gradients generated. Alright, so not only will this affect electron transport, but it will affect ATP synthesis downstream because, again, this synthase relies on the flow of hydrogens down F0. Right, oligomyosin is going to inhibit the F0 complex where hydrogens flow through. That's why it actually has its name F0, because the O stands for oligomyosin. Okay. Now, also listed here, I'll circle them in blue, are these uncouplers where we have that DNP. Now, oops. When DNP is an uncoupler, you can still have electron transport, but no longer do hydrogens flow through complex 5 or ATP synthase. They're going to pass through the gradient, through the membrane by a different mechanism. Right? So DNP, which we saw in the previous slide, the 2,4-dinitrophenol is an uncoupler. You can still have electron transport. So let me just show this to you graphically. So here we have our O2 consumption. Here we have ATP synthesis. And here we have time. Okay, so I'll do this again. So in blue, we have O2 consumption when we have succinate, ADP, plus inorganic phosphate. Then, if we add oligomyosin, which is going to inhibit F0, which is part of your ATP synthase, you're going to have a steady decline, okay, of O2 consumption. If we were to do this for ATP again, so you'd see if we have succinate, ADP, and inorganic phosphate, you have ATP production. If we add oligomyosin, we shut off ATP synthesis. Now, let's add DNP here. If we add DNP and uncouple, we can have O2 consumption again. Because the hydrogens are going to still be able to flow down the gradient. They're just going to come in through DNP, so you're not going to generate too much of a potential. However, ATP synthesis will not increase. So this is where we have uncoupled the reactions. Just to kind of drive home this point a little bit more. So, when we have electron transport, you pump 10 hydrogens out, and then they come down the gradient through complex 5, or ATP synthase. When this process is coupled, you need to have this flow of H+, or the gradient, or the potential, becomes too high, and you can no longer pump protons out. And that will shut down electron transport. If you uncouple and you provide a different means for H+ to re-enter, okay, then you can have electron transport without ATP synthesis. Finally, we're going to look at thermogenesis. Thermogenesis occurs when we have expression of UCP1. This is uncoupling protein one. Now this occurs in babies, in hibernating animals. You find expression of UCP1 in brown adipose tissue. It's brown because you have a lot of mitochondria, so it's like the dark meat we discussed when we started talking about electron transport. Now, you still have the electron transport complexes pumping hydrogens out into the inner membrane space. Okay, but instead of the protons flowing through the ATP synthase complex, if UCP1 is in the membrane, the protons are going to flow down through UCP1, and your redox energy is going to be diverted to heat production. So this is how babies in their first couple of months, they stay warm. And as babies grow, they lose their brown adipose tissue. Hibernating animals have a high concentration of brown adipose tissue, and so they use this mechanism to generate heat while they are hibernating. Right? So uncoupling can be used, for generating heat in thermogenesis. So that's going to conclude this lecture, and it's going to conclude our discussion of Chapter 19.